Okay. Thank you. Thank you so much for that really nice introduction. And I just want to thank the organizers, especially Onesmus, for reaching out to me and the prior discussions that we had. As I was mentioning, it's the first time that I'm really having a direct interaction with this group, which is a very successful group, a group that I've always wanted to work with. So I'm really grateful for the opportunity. I'll go right into the presentation. My presentation is going to center around most of what I do, which is on dryland crops and developing genomic resources for it. And just to give a quick introduction about ICRISAT, if anyone is interested to know more about the organization where I work, you can go to ICRISAT.org and find a little bit more about what we do. We are focusing on dryland crops, which are also very nutritious. The major crops are sorghum, finger millet, palm millet, and then for legumes, it's groundnut, chickpea, and pigeon pea. If you're coming from the US, it's peanut. So those are the main crops, and the map down here just shows regions across the globe where we've released varieties. And we try as much as possible to ensure that the research that we do go down or trickle down to the smallholder farmers and help improve food security. So with specific respect to dryland crops, why we work across dryland crops, obviously, we know that because of climate change, we are getting more and more regions that are receiving less rainfall, but also even those that have always been dryland. We know that they're facing challenges with degradation of soils. Recently, there was a launch of this VAX opportunity crops, and the focus is looking at underutilized crops and how they can be utilized to enhance productivity across different types of soils that are already degraded. So there's a lot of attention that is now being put into dryland crops and how we can ensure that there's food security despite the challenges of climate change. And because our focus is across mostly Asia and Africa, we know that these are the two regions that are facing a lot of climate effects. If you look at this graph, looking at temperature changes across Asia and Africa, so you can see how there has been quite a number of significant increase in temperature changes across these two regions over the last 2 decades, which is alarming. So if you see down here, between 1961 and 1972, it was more or less stable, and then it started rising over the last 2-3 decades. And now it's really on the upward trend. And it means that we really need to do something before it gets worse, that we are not able to produce at all. So both in terms of looking at crops that can endure extreme climates, but also looking into solutions that help us not to worsen the current increases in temperatures that we see. And so some of the traits that we focus on at ICRISAT is just looking at the obvious one: heat, drought, salinity. These are the obvious traits that we expect are going to worsen with increasing effects of climate change. Flooding, resistance to certain pests and diseases. You know, we know that now there are more outbreaks of things like fall armyworm, things that we didn't have before, but with increasing temperatures, we're seeing those kinds of effects. And generally just looking at how can we reduce the greenhouse gas emissions that are exacerbating the situation. In terms of the tools that we use, basically we're just looking at how to enhance or facilitate the process of developing more climate resilient crops. And the pipeline that we use is just generally coming from germplasm characterization, looking at screening, and then using genomics to look at trait discovery of these resilient traits, and then we go into validation and integration into backgrounds that farmers prefer, and then releasing the varieties and also tracking the adoption process. Now we do this together with several partners from both the national systems, but private sector as well. We also work with donors, so all our activities are donor funded, and we've been privileged to work with some of the most generous donors. And we try as much as possible, especially focusing on areas in Africa and Asia, where we could still do with a lot of capacity building, both infrastructural and also human capacity building. So most of the research that we do, we couple it with training. So training young upcoming scientists, but also training staff who are already working but are trying to strengthen or sharpen their skills on specific topics. So some of the prerequisites in terms of if we want to start a trait discovery process, we want to use genomics to enhance a particular trait. I've just put here a summary of some of the things that we absolutely must have to ensure that our discovery pipeline is going to be complete. And so traditionally, we actually worked with minimal genomic resources, but now we have so much advances in the generation of next generation sequencing data. And so for that reason, we've been able to generate reference genomes for most of the crops that we work with. They may not be like platinum scale, but we have, in most cases, the basics that we need to be able to do, high throughput germplasm characterization of gene bank material, breeding lines. We are able to screen for traits of interest and associate that back to certain regions in the genome. We're able to then validate it further, either using biparental or multi-parental populations, or just go further into genomic characterization that helps us to identify the specific genes or the specific areas where the mutations are. And then we can pass this on to functional characterization or looking at expression studies, part of what later on Hilde will be talking about, especially in what she does in the research that she is doing at the moment. And then we go back to genotype, phenotype characterization. In the past, we've come from like 30 years ago, there was more phenotypic characterization that was being done at the expense of genotypic characterization. Then we got into an era where we ended up doing more genotypic characterization and leaving behind phenotypic characterization. But there's need to be a balance in both, and especially phenotypic characterization. I think it's one area that has been underestimated, but we do know that absolutely for us to do good trait discovery, we need to have really thorough phenotypic characterization alongside the genotypic characterization that we do. And so I'm going to just go through examples, some of the things that we're doing at the moment. As I mentioned, initially we started with like basic genomes, but because we have tools available, we've gone into now working with pan-genomes that helps us to identify structural variations. So we're not just looking at point mutations, but we're looking at deletions and insertions in small regions, but also even in larger regions that could be associated with traits. And we find that this is quite the phenomenon, especially when you're looking at climate resilient traits. And then we are now in the era where we've started looking at super pan-genomes that are going all the way even up to species level. And you can look at differences that have resulted in certain kinds of evolution patterns. And so we're trying to exploit all these high-end tools in a way that is applicable to the farmer. It's not just for the sake of generating all these super data sets, but in terms of generating them to apply them for genomic characterization. And traditionally, what we've used is looking at genotyping by sequencing to look at association of markers or certain elements with traits of interest. But we're getting to the point where we're looking at whole genome resequencing because it's becoming really slightly cheaper. And the example I'm giving here is just, like about 10 years ago, it was such a huge deal for us to use genotyping by sequencing, which was only like 0.5x coverage of the genome, and we were able to discover significant things. But now we are at the level where we're doing 10x and looking at thousands of genomes or genotypes to help us with faster discovery and validation of genes of interest. When we look at even underutilized crops, where sometimes, the challenge has been in the past that we are not able to look at high throughput data sets. But I'm just giving an example here where there have been data generated for really small millets. Some of these you may not even have heard of, like, okay, so foxtail is probably very well known, but other small millets like barnyard millet, proso millet, kodo, little millets, but we are able to generate quite a bit of data sets that would allow us to do trait discovery at whole genome level. So I'll give a few examples of some of the traits that we've worked with to look at trait discovery, especially for climate resilient traits. And we typically do this using diverse germplasm, but also in biparental. And towards the end, I'll give an example of where we've used biparental population over and above diverse population to cross-validate, but also to confirm some traits of interest. So in the next slides, I'll just walk you through some of these traits. So drought tolerance, for example, one of the crops that we work with that is highly drought tolerant is pigeon pea. It's a really beautiful crop to watch in the field when there's drought, because oftentimes if you live in areas where pigeon pea is grown, you can drive, when it's completely dry, you can drive long distances, and the only things you see in the field is pigeon pea. And so we were obviously interested in, part of the group that the work that we do at ICRISAT, we were interested to know, what is it that is actually making it that drought tolerant. So in this particular experiment, the example I'm giving here, my colleagues looked at 292 pigeon pea genotypes, which are actually a representative set. So we are maintaining pigeon pea genotypes that are diverse in our gene bank. And what we've done is we've come up with a representative set of what we have in the gene bank, and they screened it, and they were able to actually identify haplotypes that were associated with drought tolerance. And so in this
I think we lost her.
Yes. Did you say
Damaris,
something?
Yeah, we lost you for a little moment.
I'm so sorry about that. I hope I'm back.
Yeah, you're back. No problem.
Yeah. Okay. So we were able to identify different haplotypes and some favorable haplotypes that are associated with drought tolerance and resulting in higher grain yield despite drought. Another example I'm going to give, and this is relatively recently done, I did mention at the beginning that most of the crops that we work with are also high nutrient crops. And so in this particular example, we looked at a variation for micronutrient content, and the two micronutrients that we're looking at was iron and zinc. And we used, again, another reference representative set, and we were able to see really nice variation for these two traits. That means there's potential to enhance them, but we were also able to identify interesting associations with both iron and zinc that now we are currently validating to be able to use it to introgress or to enhance micronutrient content in sorghum in general. Again, another example I'm going to give here is heat tolerance in pearl millet. So pearl millet is known as one of the most thermotolerant crops. It's able to thrive, well, not really thrive, but to survive under temperatures of even up to 42 degrees Celsius. And this is despite even at a reproductive stage, genotypes are able to still produce really successfully. So it's a model crop in terms of heat tolerance. And this study that I'm sharing here is we used a panel of about 340 genotypes that have been known to, they are also quite representative of what we have maintained in the gene bank, and looked at different regions. In this particular case, we actually used a pangenome, and we looked at structural variations that were associated with heat tolerance. And we were able to, well, this group, the group that did the work, were able to identify a particular region on chromosome 5 that seems to be associated with heat tolerance. And that means then we can actually just target this region and ensure that all the varieties that we develop have that particular region that is going to enhance heat tolerance, which is very much needed in most of the regions where palm millet is grown, both in Asia and also in Africa. Another example I'm going to give here is in terms of earliness. So earliness is a very important trait for drought tolerance, because crops that are early are able to avoid drought. They're able to mature within a short time before drought becomes too heating in the field. And you can see in this particular study, colleagues were able to use multi-parent populations. So it's a magic population that was developed using 8 founders that are being used in the breeding program. And as you can see, they were able to identify a really, really convincing location on chromosome 8 of chickpea that is associated with earliness. And this means then we can target this region and introduce it into varieties that we release to ensure that they mature early over and above all sorts of desirable traits that they may have. Another good example that I'm sharing here, this was published recently by colleagues from the same institute, where they looked at proso millet. So this is a relatively small millet, and they had characterized it over different seasons, and then looked at association with inflorescence length. So inflorescence length is quite important in proso millet, because it kind of defines the number of seeds, grains per head that you end up with, so indirectly contributing towards yield. And so they were able to identify some really interesting regions that are consistently associated with inflorescence length. I think this is going to be the second last example that I give, and this particular one is on disease resistance or disease tolerance in groundnut. So groundnut has one of these diseases that are very, very specific to Africa. It's called groundnut rosette disease. This work was done by a PhD student who has since graduated, Esther Echola, and she looked at, so what we did was we collected groundnut breeding lines across 6 African countries and created a reference set, a set that was representative of what is grown in Africa, and then used that to identify interesting regions that might be associated with groundnut rosette disease resistance. And what I didn't highlight is, groundnut rosette disease is found in the green belt that you can see on that map of Africa. So all that region, and including Madagascar, is affected by groundnut rosette disease. But this disease is not recorded anywhere else, and it's caused by a virus. And so what Esther did was quite groundbreaking. So she looked at, she phenotyped this representative set across different locations in Uganda, so far just in Uganda, but we are planning to do something similar across the different regions that are affected by this disease. And we genotyped the same population, and we were able to identify very consistent regions that are associated with the trait. And we went further in this particular study and also looked at different haplotypes, which was very interesting, because we also were able to find specific haplotypes that are consistently associated with the resistance. And so now what we're doing is developing markers for those particular haplotypes and using them to introduce resistance of this disease to varieties that farmers prefer. Now, the final one that I'm going to give an example is on a trait that is very unique. And this is called biological nitrification inhibition trait. This trait is actually very common in cereals, and I think there are now a few other crops that it's been reported in. And we know that when you apply nitrogen to the soil, most of the time, not all of it is taken up by the plants. So some of it is converted into different forms of nitrogen that are either leached and we lose it under, it ends up contaminating underground water, or it can be converted into forms that end up being released as greenhouse gases. And N2O, for example, is one of the most powerful greenhouse gases that can stay in the atmosphere for a really, really long time. And so this particular trait is about the natural ability of certain plants to produce nitrification inhibitors. Now, that means then the nitrogen applied or that is available in the soil remains available for a long time, and plants can take it up. So it's not converted into forms that will contaminate underground water, or it's not converted into greenhouse gases. Now, in sorghum in particular, sorghum produces an allelopathic hydrophobic compound called sogoleon, and it has been shown that this sogoleon compound accounts for up to, in some cases, up to 90% of the exudate that ends up inhibiting nitrification. And so we're really interested in looking at, what controls the production of sogoleon. And so what we did initially was look at a diverse set of genotypes, again, representative of what we have in the gene bank. And then we found these two genotypes, which are breeding lines, were contrasting for the trait. So PVK801 has relatively high sogoleon production, while 296B has low. And so we generated a biparental mapping population and advanced it up to F8, and then went ahead to genotype and phenotype for the trait. Now, as you can see, we were able to see that this trait is it's quite quantitatively controlled, and the heritability was also relatively high. That's one of the ways that we know that there's genetic control for the trait, and a good sign that you can go ahead and map for it. And so in this particular case, we were able to map for this trait. We found a really interesting peak on chromosome 7, and that's what I'm showing here, these two peaks that are controlling sogoleon production. And so we went ahead and looked at, in fine detail, the specific genes of interest. And I won't have time to explain most of what we found here, but we were able to find two interesting clusters that had interesting candidate genes, and we've gone into looking at the candidate genes as well. But we also went back into other studies that have been done looking at nitrogen use efficiency, and just looking at how well some of these crops, sorghum crops, are able to utilize the little nitrogen that you have there. And this is a really important trait, both in high intensive input systems where a lot of nitrogen is added in the soil, but it's also very relevant in low input systems, because in most places in Africa, farmers are not even able to afford fertilizer. And so they're depending on, intercropping or just using very low amounts of nitrogen. And so they are expecting that, this nitrogen is not just going to get lost through leaching or, converted into greenhouse gases. They expect that the crops that they grow should still be able to take up the nitrogen. And so we looked at other studies, and we found that the same location where we were able to identify these peaks that are associated with BNI, Also, there were independent studies before that had identified that region to be associated with nitrogen use efficiency, which is interesting. And so some of the candidate genes that we found there, we are currently using them for, just characterizing them further to see how they are involved, whether in the regulatory pathway, how they are actually involved in the production of sogoleon in sorghum, and how we can exploit that to ensure that, when we release varieties of sorghum, we are actually releasing varieties that are more nitrogen use efficient. And so we are not just applying too much nitrogen that gets wasted, but we are applying the minimal that the plant needs to sustain it and still get good yields. So in summary, we do have abundant opportunities that exist for dryland crops that we are currently testing, and we see quite a number of traits that still remain unknown but are very interesting. And even though there are some things that we can learn from major crops, there are also things that we just have to do for these dryland crops, because those are things that are very unique for them. And as I mentioned already, we need to invest both in phenotyping and genotyping if we're going to end up with regions that are actually contributing to the traits of interest. We're looking to collaborate more across different institutions that have different expertise that we can utilize in a more efficient way, both to capacity build young and upcoming scientists, but also just to help in the process of identifying all the interesting traits that we would like to improve the resilience of these crops. And in the next talk from Hilde, you're going to hear more about things like looking at functional characterization of genes and how it can be exploited. And we want to get to a place where our discovery pipeline also goes all the way to functional characterization, so we are able to use all the different tools available to enhance resilience of such kinds of traits. And that's it for me. Thank you so much. I just finished by thanking everyone who has supported us or who has collaborated with us. I couldn't write everyone here, but these are just some of them whom we've been working with. And just want to thank everyone for contributing to the work that I've presented here, and to thank you all for joining in and listening to my talk. Thank you. Thank you so much.
Thank you, Damaris. Thank you very much for this very nice talk, showing how genomics can help discovering a lot of information and traits for drought and other. Oh, I think I was muted, sorry.
Yeah, we lost you for a minute.
Okay, so anyway, thank you. Let me take some questions from the chat and from people that will raise their hands before we go to Hilde's talk, and then we can have like some more common questions that we can both answer. So let me start with the first question that was asked by Joel in the chat. Do you have a phenotyping facility? So basically, are you doing phenotyping in the lab, or are you doing everything in the field?
Yeah, that's a really good question. We do both. So in certain cases, we are able to do phenotyping in the lab, but in most of the cases, we are doing in the field. For drought, at our headquarters in India, we also have like a rain-out shelter that we're able to do, because for drought, sometimes it's really difficult, because when you've set up your experiment to do the drought, that's when the rain pours. So it depends. It really depends on what we're doing. Things like looking at root traits, those we do in the lab. But the things like the groundnut rosette disease that I showed there, that we've all done in the field. So we do both. It depends on the trait that we're working with, and we could strengthen our phenotyping. It's not always great. It's quite a bit of a challenge phenotyping well for climate resilient traits. So we could strengthen it, but phenotyping is also one of those things that is get really, really expensive. But something like looking at micronutrient content, that we can do very comfortably in the lab, and it seems to work really well. So it depends, in a nutshell, it depends on the trait we're working with.
Okay, thank you. Another question on the sorghum, the Sorgo-Leon story. Is this similar chemical also present in other cereals, or is it like sorghum specific?
Yeah, so the trait is not, so biological nitrification inhibition trait is not specific to sorghum, but the compound Sorgo-Leon is specific to sorghum. So you have different compounds in different crops. So for example, in maize, they've identified a different compound, in wheat, a different compound. So you get different compounds in different crops. But Sorgo-Leon is very specific to sorghum. It's also relatively easy to phenotype because it's hydrophobic. So, you just look for the oily substance. The pathway is also really well characterized. So Sorgo-Leon is very specific to sorghum. Yeah.
Okay, thank you. I see one question that I actually expected. How can you help farmers in the applicable way?
Yeah. And so that's some of the things, and I'll just stay with this slide to explain how we would help farmers in an applicable way. So the example I was giving of farmers let's say in Africa, most of them, farmers who grow sorghum, they do not add any input. So for that reason, most of the yields are quite low. Now, how would a trait like this one help? If we are able to ensure that the varieties that we end up giving the farmers are more nitrogen use efficient, they are producing high levels of Sorgo-Leon, and for that reason are not ending up having the nitrogen that is applied or is available from the atmosphere just get leached out, we will be able to enhance their yields without necessarily doing much. So they're able to utilize the little nitrogen that is available in the soil to improve their yields. But one of the traits that are really, really troublesome in African locations is Striga hamontica, which is a parasitic weed. This weed actually devastates most sorghum, most cereal fields in Africa, depending on the region where you grow it. It has been shown that sorghum genotypes or cereals that are more nitrogen use efficient suffer less from this parasitic weed. And so this is a practical example where you're putting this into the farmers' varieties, and so they're able to reduce the effects of some of the things that really affect them. But this trait is also applied in high-intensive production system. We have regions in India, for example, where farmers are really subsidized, and they usually put more nitrogen than they should. And for that reason, you have a lot of contamination of underground water systems, but also you have a lot of greenhouse gas production. When we use BNI sorghum, for example, we see that even up to 3 repeated seasons, farmers don't need to apply more nitrogens, because you find that they still, after 2 seasons, you still find that there's nitrogen that the crop can take. And if you compare the BNI sorghum versus non-BNI, you can see just after 1 season, the crop is yellowing. So that's one practical example. And I mean, I can go ahead and give more and more, but just staying with that one, that's very practical, because we are introducing these traits into farmer preferred varieties and giving farmers traits that they are able to use. So, I mean, like even for drought tolerance, if you're able to introduce earliness, then the farmer is able to get some yield within a short time before the drought comes and devastates everything that they have in the field. I hope that is convincing that we're doing something that is practical to the farmer.
Thank you. There are many questions popping up, and I will keep some for the end, but I would like to ask you a last one before we move on to Hilde's presentation. And it's a long one, but it comes like you have been doing GWAS on several crops, on drought and so on. You have been showing nice pieces of chromosome or loci that were responsible. So the question here is like, how does those drought genes interact? So basically, how many genes are involved on drought tolerance? How do they interact? Do you have like more precision in that? That's, I guess, the question. And the last, sorry to interrupt you, the last thing is like, does epigenetic have influence there on these genes? Thank you.
Excellent question. And all of the questions that you've asked so far were really, really good, and I've enjoyed the responding to them. Yes, most of the climate resilient traits are quantitative traits. So we end up finding quite a number of regions in the genome that are contributing to the trait. So, and most of them have small effects. So ideally, the most ideal way is to improve all of them, because also there's interaction between them. And the examples that I've given, especially for the biparental one, where we're looking at sorghum production, is just one particular locus. And I wanted to give this example to go towards, just as a segue into what Hilde will be talking about. But in most cases, there are different, several contribution from different parts of the region of the genome contributing to that trait, and there's interaction as well. And so actually the most ideal thing that we're doing right now is introduce all the markers that we have that are explaining at least 10% phenotypic variation. We put them into a genomic selection panel. And so we are able to select for several different things at the same time and using them to predict performance. And so that gives us more room to play with, not just one trait, but also several traits. So we are able to look at interaction, because I mean, in the field, you don't just want a drought-tolerant variety. You want high-yielding, drought-tolerant, disease-resistant, all those many things. And so looking at the interaction of all those, even as we try to make sure that we're introducing these into the right backgrounds, we do it in a way that we are able to select for all the different things at the same time. And so what we do typically is when we do trait discovery, even though we might find just one major trait, okay, if it's a major trait, it's relatively easy to look at the interaction in the different backgrounds. But if we find quantitative traits, which is the case for most of the resilient traits, we put them in a genomic selection panel that allows us to also look at interaction across the different traits by themselves, but also interaction with different backgrounds that we work with. Now, we expect that there are epigenetic effects. A lot of studies have shown, like even stress, any kind of stress has an effect on the epigenome. Now, specifically looking at trait discovery, we haven't exploited that area that much. And, that's one of the areas where we would be looking at collaborations to look at, how does the epigenome affect some of these traits, both in individual backgrounds, but also across different backgrounds. Is it heritable? Can we introduce the associated regions, epigenomes into the genomic selection panels that we're using to improve our prediction accuracy? So thank you. Thank you so much for the very, very interesting questions that we've had. Yeah.
Thank you, Damaris. Indeed, the chat is very active, so that's very nice to see. And we will have some other questions at the end, I'm sure, for the next discussion. So our next speaker is Hilde Nelissen. Hilde Nelissen is a group leader at VIB here in Belgium, the Center of Plant System Biology. Her research career has focused on the central biological question: how do growth processes determine final plant organ size? Initially, she approached this using molecular biology, studying Arabidopsis leaf development, but her interest shifted towards more applied research, and her focus thus redirected to maize. What started as translational research to bring knowledge from Arabidopsis to crops and from the lab to the field gradually developed into a research line with the goal to decipher the instructor networks that govern leaf size, organ growth, and ultimately yield in maize. Because plant organ size control is an important yield component that is also impacted by adverse conditions, the ultimate goal of Hilde's team is to deepen their understanding of the growth regulatory networks to achieve climate-resilient crops. Hilde is co-chair of the Plant Cell Atlas Committee for Spatial Omics, adjunct director technology transfer of the Center of Plant Systems Biology, and professor at Ghent University, where she teaches plant yield, plant research technologies, and academic storytelling. Hilde, your presentation is on, and the floor is yours.
Okay, thanks a lot, Mark, and especially also Onesmus to put this nice program together. I must say that Damaris already put the bar quite high, so I will try to live up to what she told us. So our team actually focuses on understanding growth processes. And as you can see from these GIFs that were taken from maize plants that are growing on our automated phenotyping platform, when we take a picture every day, you actually see that continuously growth is taking place throughout the development of the plant, either within one organ that keeps to grow or within the production of new organs in that growth. And when we try to summarize this and actually sum up the growth of all the leaves, all the internodes, and then the ear, we actually see that this happens in a highly coordinated way. So whenever the leaf growth is maximal and starts to go down, the plant starts to invest in the growth of the internodes. And similarly, when this growth is maximal and goes down, only then the plant starts to invest in ear and the reproductive organs. So this is under control well-watered conditions. And so what we use drought for mainly is as a way to perturb our growth processes. So we don't use extreme drought, but however, we use actually very mild drought conditions. And if what we can see is that for all the different organs, these drought conditions lower the growth rate at which these organs grow. They prolong the duration at which these organs are growing, but they do not affect this coordination between leaves, internodes, and ear. And this means that there is actually a very tailored mechanism taking place that actually allows to do growth under drought conditions. And that's what I'm going to talk to you about today. So I showed you the mature plants, but actually the major focus or the working horse of our team is looking at maize leaf development, and particularly during seedling growth for several reasons. First of all, because we see the same mechanism as we saw in the mature plants. So we have a maximal growth of the leaf, and that goes down at a certain point, which is the moment when the next leaf starts to develop. And under drought, we also see that we get a reduction of the growth rate, but a prolonged duration of growth. So we really think that this leaf we can use as model to simulate how all other growth processes also in mature parts of the plants are taking place. Now, what is very neat about this leaf, if you tease it out from the shield that is formed by the older leaves, then actually such a growing leaf is very nicely organized in a spatial way, in the sense that the dividing cells are at the bottom of the leaves, then we have a portion of expanding cells, and then we have mature cells. And since the maize leaf is quite large, this allows us to sample sufficient input material for a lot of the molecular analysis that we can do. And so we can do this on enriched enrichment for dividing cells, expanding cells, and mature cells. Now, what we also see is that these zones, they are not stable. They fluctuate over time. And with this, I mean that this division zone or this portion of dividing cells, that stays maximal during the time this leaf is maximally growing. But we notice that from the moment the leaf stops growing, this pool of dividing cells also starts to decrease. So there is not only a spatial, but also temporal regulation of growth. Like Mark already said, we originally started this research with a translational hat on because a lot of information was available on Arabidopsis leaf growth, which has the same organization in space and time of these growth processes. And so we wanted originally to know how many and which of these processes that were identified in Arabidopsis also hold true in maize. And so with this, we collaborated with a team of Dirk and Zee, who really studied leaf growth in Arabidopsis, and they came up with all these complicated networks. So they identified many players, of course, also together with colleagues in the field. There were connections between the different genes. And so we tried actually for as many genes as possible to find the counterparts in maize and to actually make the same modulation. This could be a mutant, an overexpression, a specific overexpression, or so on. And then we evaluated whether or not we saw similar phenotypic outcomes in maize as was seen in Arabidopsis. And I can cut here a very long story quite short, but if you want, I can always elaborate on this. The majority of this, as we already know, did not translate. However, there were some modules that actually do translate, and due to the sake of time, I only have time to highlight one of them, which will come back later on in my story, which is actually a story of a cytochrome P450 family, which in Arabidopsis is called CLU, and in maize is called plastochrome 1. So if you look at plastochrome 1, it actually has growth phenotypes because the leaves are shorter than those of the control plants. But what we see is that there are more leaves made in the same amount of time. And this is why it was called plastochrome, because this is the time in between the different leaves that are developing. And from the expression profile of plastochrome 1, we really see that this takes its action in the shoot apical meristem. This is another part of the research, is a deep dive in this molecular function of these key players. However, again, for the sake of time, I cannot really elaborate on this today. So, but plastochrome 1 in Arabidopsis was shown to have growth stimulatory effects when it was higher expressed. So naively, we originally constitutively overexpressed it. And as you can see from the leaves of this plant, this was a primary transformant. You see that there are gigantic leaves being formed that actually alter the way a maize plant looks compared to mock control that was in the greenhouse at the same time. So this really shows overexpression of plastochrome 1 enhances growth, but actually it is not really what we want, and it's also not viable to go to the field. So then we went in and we took another promoter, which was less active. We just stimulated the expression of plastochrome 1 a little bit further into the emerging leaves, which was actually a GA2 oxidase promoter. And when we use this promoter, we could still see the growth enhancement, but it was much less than when we did it constitutively. We saw very nice traits in the greenhouse, so we took this out to field trials, both in the US and in Belgium, and we could see that there was a positive effect both on biomass and on seed yield present. And so this really shows the example that for a lot of these agronomic traits, we really need to move away from constitutive perturbations, and we need to go towards fine-tuned perturbations. Now, when we look really into which processes that were affected in these positive plants, then we actually see that this was against cell division. This is the size of the division zone in the non-transgenic siblings, and this in the transgenic ones, and we can see that there is a slightly larger pool of dividing cells, but what is most remarkable is that it remains active for at least one more day compared to its non-transgenic siblings, and that this is actually causing the growth benefits that we see happening. So what is now actually the role of this prolonged duration of growth and this prolonged activity of the cell division zone? So we saw this under drought. Again, here you see under well watered, we see the leaf in white, the growth rate. We see this in drought in the black square. So it is lowered but prolonged for a longer period of time. And so we were wondering, what is now the biological role? Why are plants doing this? It feels like they are making themselves smaller, arresting their growth a little bit. And we were hypothesizing that this was because they were waiting for the stress to be relieved so that they could resume their growth. And this is, we tested by using our drought system, and we re-watered at different time points during the growth of this leaf. And what we saw was actually very remarkable. When we re-watered, which is always the red arrow, in the beginning of the growth, then we see that the plant can completely resume its growth rate up to well water levels, and we hardly see any effect anymore on final leaf length. The similar thing was seen when we applied the re-watering a little bit later during the growth, when the division zone was still active. When we do this, we still see that plants are able to restore their growth when the stress is relieved. This was less the case when we started re-watering at day 6 here, or even later when the cell division zone is already decreasing. This means we can still stimulate it a little bit, but this is only due to cell expansion, not anymore through cell division. And so this actually means that this prolonged duration of growth seems to have a very beneficial role, especially these days in this changing climate, where we have longer periods of drought occurring, but then all of a sudden water comes available again. So this is really, I think, an important trait in the changing climate to follow up on. And so we then also showed that with lines that have a prolonged growth, like the ectopic expression of plastochron 1, when we there performed the re-watering experiment, we actually could see that this line had much better potential to recover their growth upon re-watering for a much longer period of time than was seen for the non-transgenic siblings of this line. And so this really shows that this prolonged duration of growth had some important features. We did this experiment not only in the reference genome B73, but we also did it in a recombinant inbred population. And so we saw that this was indeed under genetic control. We had now one player, so we were interested whether or not we could find novel players in this process. So we did 2 different approaches. One approach was doing a bulk RNA-seq at many different time points during drought and re-watering. And at the same time, we also applied a single cell experiment upon a short drought and a longer term drought. And actually what was remarkable was that we found the same gene that popped up as really a big candidate, was a histone 1-like candidate. So we were so convinced that this gene could have a role that we decided to use this again for genome editing. And what we could see, we got actually 2 types of mutations. We got an in-frame deletion, which is the one that you see here depicted next to a wild type from the same segregating population. And so we could already see that it has growth effects, it's smaller, but we also see that this line is much more sensitive to many different stresses, amongst which drought. We also found an out-of-frame deletion in the linker domain of this H1L, and those plants seem to be more tolerant to drought. When we do this in our phenotyping platform, we see that we can increase leaf yield, leaf growth slightly, but we also got a positive effect, for instance, on stem biomass. So we are evaluating these lines as well in field trials now. So this is one example. This is one gene that really popped out from this transcriptome analysis. But of course, when you've ever done a transcriptome analysis, you find many genes that actually would be potentially interesting to functionally validate. And if we need to do this one by one, this is still quite a tedious process, like we can see here for H1L, because not all of them really work always very well. And for this, we actually use now a multiplex genome editing strategy in the lab, which is called BREEDIT, which is an acronym of breeding together with editing. And this is a way that allows us to narrow down gene space. So if you have many different candidates, we can test them in making populations. I will explain in a bit how we do this. By making populations, we can immediately find the most interesting candidates to work further on. But on top of this, this technology also allows us to assess which combination of edits would even enhance certain phenotypes or certain traits. So what we actually do is find many different target genes. And as I said, in this case, they came from transcriptome studies. So then we start to design guide RNAs. And in this case, we did this for about 60 genes. And we cloned then the guide RNAs for these 60 genes into 5 different vectors, which actually means that each of these vectors contains guides, 12 guides that each target one gene. Each such script or vector was then transformed into a maze line that already carried Cas9, the nuclease. And so we generated populations, for instance, of script 1, in which 12 genes, up to 12 genes were edited. Not always all genes were edited. Sometimes we had a combination of 2 genes, 4 genes, sometimes only 1 or 2 genes were edited. Now, where does the breeding come in? We now generated small populations, which on their own we can already phenotype and genotype. But the neat thing is actually that we now can also start to combine high-performing plants, for instance, from the script 1 population with plants of the script 2 population. So now we don't only have 12 genes, now we have 24 genes that are segregating in this population. Now, every population is thoroughly screened. With this, I mean that we phenotype it for as many traits as possible, but we also genotype every plant. We do this by multiplex amplicon sequencing of all 60 genes in all different plants. And so what we then start to do is make simply set, but it's more complicated than that. Simply set, we make associations between the phenotype that we see and the genotype. And more recently, we are actually also developing a machine learning or an AI model to do this for us. But the outcome of such models are actually networks like these, where we start to get certain traits, and the model then explains whenever a certain gene is mutated, it has a positive or a negative contribution to a certain trait. So when we did this for the 60 genes that we picked up in this transcriptome study, we added some positive and negative controls, but in total, we could pick up about, I think, yeah, something is blocking here the screen. We had 80% of the new targets we could associate to a drought phenotype, which actually was either positive or negative. And this really gave us a shortcut. And so now we have a set of target genes that might have novel function under drought that we are now drilling deeper to understand the molecular phenotype of these lines. Now, I will shift gear a little bit to make an introduction to the booster project, which we are part of. And this will seem like a break, but I will come back later to it. What is our role within the booster project? And it's exactly that, applying again the genome editing and preferentially multiplex genome editing to this project. But first to explain the project, so it is an EU project with about 14 partners from 10 different countries. And it actually has a very dual approach to come to better improved drought tolerance, mainly of maize. But we also use different types of crops that have a better drought tolerance than maize. We do this at 2 different levels. So the first one is at a genomic level. So we screen mainly European germplasm for maize and also for teff to understand what is driving the drought tolerance genotypes. And in a second strategy, we are using dry soils from dry lands, like Damaris explains, to understand biostimulants that live in there. So to identify, for instance, PGPR, plant growth promoting bacteria that are already present in dry soils and that could have a positive effect on maize, but also on teff, so that we can actually also develop biostimulants. And then these two are, of course, not separated. We also would hope that towards the end of the project, we could examine whether or not there is a synergistic effect between the 2 different strategies. So I hope from my previous explanation that you understand that I am mainly involved in the first part of the strategy. And the strategy that we take there is based upon a technology that was spearheaded in the lab of Thomas Hartwig, who is also in our booster consortium. And he developed what is called the MOA-seq technology, which looks very much like an ATAC-seq technology, which means that it goes and it looks for fingerprints that are actually accessible in the chromatin, and this is because they are bound by transcription factors. I'm not going to go into detail exactly how the MOA-seq experiment works, but I do want to highlight that he has found a very neat way to identify regions within a natural population. Because in the past, we've also done projects where you have a drought tolerant and a drought sensitive maize line. You apply drought to both of them. You sample, for instance, for RNA sequencing or for any molecular technology, and you find a huge amount of differences between the 2 varieties. And it's very difficult to find the ones that are causal for the drought phenotype for which you actually started. What Thomas now does is make actually a hybrid between those sensitive and the tolerant line and subject the hybrid to the drought. By applying then his MOA-seq technology, he finds then the peaks that have accessible chromatin under drought. But the neat thing is that he can assign this allele specific, so he can say which variant is present in the drought tolerant line and which one is present in the drought sensitive line. So we really get a nice idea about which transcription factor binding sites are actually more or less accessible in the drought tolerant varieties versus the drought sensitive varieties. We don't need to worry about all the rest of the effects that are changing in the background. We also don't need to worry about the drought treatment being different because we all do this within the hybrid background. So currently, the partners of the Booster Consortium are performing this MOA-seq. They are combining it with RNA-seq. So we really learn which genes are transcriptionally drought responsive. And at the same time, we learn how their transcription factor binding a landscape in their regulatory sequences is actually looking. And with this information, that would then come to our team. There we are actually using genome editing to validate these identified cis-regulatory elements. And indeed, the ultimate aim also for us would be to actually start to exploit these best variants to actually come to lines with improved drought tolerance. Now, we are still very early time within this project. So we are doing mainly the MOA-seq within the consortium. But our team is currently setting up all the different tools that we would need to use genome editing to do this validation. So I already explained to you, we have a pipeline for the multiplex genome editing. We also want to see the oligo insertion, because we are using not only maize, but also teff, which is already more drought tolerant than maize. We are also using Nindensis, which is actually a drought desiccation plant, tolerant plant. And so we hope to also identify regulatory elements that might be present in the more drought tolerant plants that we could maybe also introduce or mutagenize within the maize plant. Now, because the technology of Thomas actually works with hybrids, we also want to do this in a drought tolerant and a drought sensitive variety. So the last couple of months, we mainly focused on also getting the transformation and the genome editing up and running in the more drought tolerant variety, because in the drought sensitive line, this we already had up and running in the past. And so actually, this is what we are currently doing within the booster project. And I hope that you then also understand that we can start to combine this. We can do genome-wide or almost genome-wide. We can start to look which are the most prominent regulatory elements that could lead to drought tolerance, and then we could start to use this multiplex genome editing to improve, for instance, a drought tolerant inbred line and move it closer towards a drought to start from a drought sensitive line and move it more towards being drought tolerant. But of course, this multiplex genome editing technology could also be used to start stacking different traits. So here we were now talking about drought tolerance, but we can also put in, for instance, more architectural traits as well. And then on top of that, to make then the link to the presentation of Damaris, there are a lot of interactions going on between smaller QTLs or smaller genomic regions that all contribute to a certain trait like drought tolerance. So also, I think this multiplex genome editing tool could also use or be used to bring together these more of these small effect QTLs within one of those lines. And I do see that there is a lot of synergy of bringing this together. And in addition, I think also genome editing could be used to access those regions of the genome that are very much linked and not as accessible for the crossing and the more natural breeding approaches. So with this, I hope that I have convinced you that the duration of growth and the size of this division zone is really important in a changing climate. We have tools up and running to do multiplex genome editing, to screen gene lists, to do QTLs, and to combine edits and traits. And we are part of the booster project that actually really wants to take lessons, not only from drought tolerant plants, but also from dry soil, and to see how we can use this to apply this for technology. Now, I'm here today only voicing the hard work of many different people. I'm sorry if I couldn't present your work here today, but the focus was on the drought work, which is now currently mainly driven by Yang and by Yasmin in our team. I also would like to thank the numerous collaborators and funding agents that show a lot of confidence in us to support us. And then finally, I would like to highlight that this breeded technology has a lot of applications, not only in maize, but also across different crops, as I said, to combine traits. And for this, we are looking to spin out this technology into a company that for now has the name Rainbow Crops, and we get a lot of help from VIB headquarters to set this up by Tom and Giacomo. And with this, I would be happy to take questions.
Thank you very much, Hilde. I will see the questions popping up very soon in the chat, and I will come up with it. but maybe I can start with something. I hear from the two talks that we had two different approaches for the drought issue. We've heard from Damaris that she was looking into short-term, short growth duration or early maturing varieties. And that's typically for a climate in the south where we have a rain season and then you need to maximize in that period. Whereas what you propose here is a long-term approach because you want to bridge basically the two more humid periods, let's say, that you need to bridge this summer that is very dry. Do you think we will have anything common that we will look at in that case, or do we have like two very different approaches?
Well, I think a bit of both, actually. I think the truth lies somewhere in the middle, as usual. I do think there is the two traits can be combined. This I strongly believe. I think what we now do is tease them apart and really focus on the specifics of one of these two mechanisms to understand them. However, I'm also quite convinced that we will find some genes or genomic regions which would be an overlap between the two, because that's also what we see if we make cross references from our mild drought and our prolonged duration set of genes with longer or with other drought studies. There are always some genes in common, and I don't think we fully understand how there is the synergy between them. But I think we now have the tools and start to get the know-how to start to address this. And I think it's actually quite interesting to see how we can bring this now together, and if possible, learn from the differences there are and see how we can tackle this in the future.
Okay, thank you. Hewan from Ethiopia is asking a question, and she says like teff and maize both possess drought-tolerant genes, but they are not identical. So a bit coming back to this. Each crop has evolved different mechanisms to cope with drought stress. So how can we use the genes from these species to develop drought tolerance? Because it's probably a different mechanism.
Yeah, that's exactly what we want to do. So we want to do some kind of comparative analysis. So you can learn two things: what is already in common between a drought tolerant and a more drought sensitive species, because as you said, both of them have mechanisms. So first of all, figuring out what is in common is one thing. And a second thing is looking at the differences and then see how can we use this. Maybe there are, we've thought about many different scenarios. We don't know exactly what we're going to find, but you can start to think about, for instance, genes or mechanisms that are present in maize but are not activated. By looking at the regulatory elements, we might find ways to actually activate them as well in maize. But you could also think that there are indeed many different pathways which are not present in maize. That is then, I think, more difficult to solve by just using a genome editing approach. So we don't know what exactly the outcome of the comparative analysis would be, but I think it's worthwhile to do it, and there are definitely learnings to take from it. And if possible, we will address it through genome editing.
Great, thank you. You've been talking in the first part of your talk about the different genes and that you were overexpressing them to find, to get impact. And so at one point you have mentioned kind of fine-tuned gene modulation. How do you approach that actually? Can you explain a bit more about this, please?
Yes, so it is clear there were some publications from companies where they massively took genes, for instance, known in Arabidopsis, and then they went to a crop, very often with constitutive perturbation. And when you go through the pipeline towards application, you see that although they are promising in the greenhouse and in the lab, they are not promising anymore when you go out in the field. There are several reasons for this. There is this translational gap between model to crop. There is a translation gap from lab to field, but there is also the fact that constitutive expression is actually, I think, not the best way forward, because you bring a gene in cells where it normally is not, it causes negative and pleiotropic effects. So I am a strong believer for the fact that we should fine-tune genes, but not only genes, but entire networks, actually. And so the way we are approaching this within our team is by using spatial transcriptomics, for instance. This is a technology that allows you, within the tissue section, to visualize the expression of many genes simultaneously. And if you do this on low-yielding varieties and on high-yielding varieties, you can actually see in which cell types, or quantitatively that you need to change the expression of a certain gene, and you hardly ever see it upregulated in all cell types. What you will see is that it is a little bit higher or in a little bit bigger zone of expression. And I think you can use spatial transcriptomics to map this and to give you then a blueprint of the map where you want to go towards and how you want to tweak genome editing. And there again, the data from the Thomas Hartwig's team, like the MOA-seq, having these regulatory elements available to tweak the expression of certain genes is highly valuable in this strategy, I think.
Okay, thank you. I don't see many questions popping up. I think we have making the people suffering a bit, a lot of molecular information. Damaris, maybe you can join us again if you want.
No, just maybe just briefly to say thank you so much, Hilde, for a really, really excellent presentation and the research work that you doing, and most of it is really relevant to what we do. And yeah, thank you and congratulations for such a great job.
Thank you, that's very kind.
To both of us, of you, sorry, there were like a few more questions, and as you can imagine, people are working on different crops, and actually like how can all that knowledge, I mean, working on what you are doing, working on what you both are doing on different crops, how can people use that knowledge for the crops that they are interested in? Is there any potential to use that data information or even the genes, let's say, that you discover in other crops? And when I say other crops, I mean, I'm talking often about orphan crops. We have many African participants in this meeting that are talking about tuber crops that were not much tackled in this presentation, but also more legume crops. So this approach, how would you see that? Let's say that you can expand it to other crops. Thank you.
Is it?
Maybe I'll try.
Both, I guess, whenever.
Go ahead, Damaris.
So in two ways. One way that I see, for example, like the kind of work that we do with all the climate resilient crops align really well with what Hilde is doing in maize. And the missing link here is that, you know, we are doing very traditional approaches. After discovery of traits of interest or candidate genes, we are looking more into introducing them directly into different backgrounds that farmers prefer. But she goes another level and looks at functional characterization and looking at how to do multiple edits at the same time. And so in that way, we can actually combine both. I think the biggest challenge we face with our crops, dryland crops, is that they're not very easy to transform. And, one of the things that we find with sorghum, for example, you optimize today your transformation for genotype X, and next time you want to introduce something into a farmer preferred variety Y, you start again. And so maybe, looking at different ways of overcoming the recalcitrance that we find in some of these dryland crops will be one. But the other thing is, now, if you're trying to work on a completely different crop, let's say it's an underutilized tuber crop, I think there's opportunity to leverage everything that we're doing, both what Hilde is doing and what I'm doing, to come from, you know, just genome characterization, understanding the gene landscape of your crop. It's not really expensive now. And then going into doing screening for the various traits of interest, understanding different genes of interest and characterizing them further, looking at the whole pipeline. Hopefully you get funding for it, but looking at the whole pipeline and make sure that these crops, even though they are not as well studied as a crop like maize, do not have to wait another 10 years to get to the level of research like what Hilde is doing, but just leverage some of the things that we've already learned working on these crops, whether it's for maize or sorghum, and then bring them together in one platform and make it work for your crop, that particular crop that you're interested in.
I agree with that. I think it's also part of what I presented is technology. We developed it in maize, for instance, the breeded technology, but I also think about the spatial transcriptomics technology. All these technologies are applicable to other species. I don't say they're always as useful in all species, but most of the time, technologies, when they are tested and de-risked, it's already in a crop in maize, then this can be used. And another thing about the genes, what I still think is cool that we start actually from this translational research. We started from Arabidopsis and we actually are continuing to work with those genes that gave a similar response in Arabidopsis and maize. Those two plant species are actually quite distant from each other. And so this means that we are working with a set of genes that actually have a very conserved role across many different plant species. And so this, I think, would also allow you to get some learnings from in other plant species. So I think both at gene level, but also at technology level, these are things that indeed you could take along, and there are already hurdles overcome. I'm pretty sure in the work of Damaris, there is indeed also the publications that she has is struggles that they had, and I think all other crops can learn from this to speed up their innovation.
Thank you. Yes, there are definitely enough technologies to help people to speed up research and applications in the field. I would like to open still the floor to questions. You can ask them directly. Just raise your hand, please, if you have a question. If not, or meanwhile, I would like also to ask you to switch on your camera if you want, so that we can maybe take a kind of group picture for this online seminar. Thank you very much. Will we see some pictures appearing? Yes, people are appearing. Hello.
Hello, Mark.
Yes.
Can you hear me? Sorry, I was able to join in late, so I just got to hear the last part of the discussion, and from what I hear, it's an amazing discussion. However, I think it's a recorded meeting, so in any chance, I'll just be able to follow it through later, and in case of any questions, probably I'll just raise them up and
yes, we will make this session available on YouTube, so you can look it afterwards again, and you can send us the questions, and we will forward them to the speakers. No problem, definitely.
Thank you.
Okay, so I don't think there are more questions. So Nesmus, I like you asked for a group picture. Can you take that? It's done? Okay, so I'm left to see in this very diverse group of attendees that I'm very happy to see like this. I would like to thank you all very much for attending this session, for actively asking questions. I'd like to specifically thank the two speakers who have given wonderful talks, I think, where we see that from fundamental research to application, we can have an impact on the ground for agriculture. And I think that most of you are interested in doing that. So I would say keep up the good science. Also, big thanks to Onesmus, who has been organizing all this and who makes sure that you are all attending this meeting, which I think is excellent. Thank you, everybody. Have a wonderful end of day in the south. Have a wonderful afternoon in the north, or wherever you are. Thank you very much. Bye-bye.
Okay, bye-bye.